David, thank you so much. Oh, there you are. Okay. Thank you so much for inviting me back to the Superheroes of Science lecture series. It's always fun to come over. Last time I was here, rain was just a twinkle in David's eye. I don't think that he even had the lease signed on this space. And so being able to come here and walk through the labs that he's setting up, along with many of you here in the audience, is an inspiration, truly. I love to see the kind of innovation that's going on in this building, and I'd love to have that spread more broadly, and I know that that's David's dream too. Let's see, I don't know if I can compete with an auction and beer in the back. I'm gonna try and do my best. As David said, I met him about 10 years ago when he was at Columbia, and we're both pretty interested in using sequencing as a way to characterize unknown pathogens in human clinical samples, and he was looking at biotech kind of ways to enable that. I was running a program at Sandia at the time that was taking a look at developing new methods to do that. My background is as a computational biologist and chemist, so as David said, a lot of sort of rational drug design type of work in my background, but also some work in protein structure modeling and prediction, and using chemical cross-linking types of methodology to get at structural information about proteins that you can incorporate into modeling approaches. Brought that to Sandia, but then quickly kind of climbed the management ranks, left Sandia running the program, and then came out to PNNL, back to the Pacific Northwest, back to where I'm only a 3.5-hour drive from my family, so it's wonderful. I titled the talk, 10 Science Discoveries You Can't Live Without, in parentheses, courtesy of the national labs, because I was pleasantly surprised, honestly, when David asked how many people know about PNNL, and there were more than like 1 or 2 hands went up. That is really unusual. And I would say that just the national lab system in general is not really well recognized or understood. And so it's a great opportunity for me to be here with you guys tonight to tell you a little bit about some of the scientific discoveries that have been made by researchers at the national labs that really have changed the way all of us live today. And so I wanted to first get started by introducing ourselves. So for those of you who have not heard of the national labs, there are 17 DOE national labs. They're located all over the country. PNNL's circle is, of course, the largest one, and it's, of course, the best, so you guys can walk away with that. This isn't being recorded, is it? Oh, it is. So anyway, the goals of the national labs are really to support the missions of the Department of Energy, and there are 4 missions: scientific discovery, national security, energy, and environmental remediation. And so all of the labs in the network have in the mission space 1 or more of those goals of the Department of Energy. I would say that PNNL, I'll tell you a little bit about PNNL. PNNL, as David said, is about a $1 billion R&D laboratory located in Richland, Washington. We have about 4,500 people working at the laboratory. We do work, actually we call ourselves a multi-program lab, because we do about 20% of our work in fundamental science. Another 20% of our work is in applied energy and environmental programs. About 1/3 of the work that we do is for nuclear nonproliferation, and the rest of the work we do across the federal government for agencies like the intelligence community, Department of Defense, National Institutes of Health, DHS, Department of Homeland Security. And so we really have a very diverse set of programs that we run at the laboratory. I wanted to give you a brief history of the National Lab System. So as many of you know, the National Lab System really started, I'll try and get out of your way here, in the 1940s with the Manhattan Project. And the goal of that project was to, of course, develop an atomic bomb to be deployed during World War II. But each decade had its own series of challenges and opportunities for the national labs. And so in the 50s, the labs were maturing. They continued to develop new technologies for the nuclear arsenal and nuclear-related science. In the 60s, they started to expand the programs out a bit more to do fundamental research. But the 70s was where the energy crisis really changed the character of the national lab system. The national labs were deployed to help deal with some of the challenges the nation was facing in terms of energy supply. And so the portfolio of programs at the national labs expanded to include energy. Over the following decades, we had times where the labs were focused on engaging the private sector. Another really large perturbation was with the end of the Cold War. And so the labs, particularly those that were involved very strongly with nuclear stockpile sort of management, stewardship, had to shift their activities significantly as a result of that change. And then immediately, in the next decade, we had 9/11. Now, I was at Sandia. I just started there. I was a few years in when 9/11 took place. I'm sure all of you remember where you were on 9/11 when the first plane hit the World Trade Center. But it changed really the whole focus of many of the national labs and expanded our portfolio into Homeland Security. I know that in the biology program at Sandia, especially after the Merithorax attacks that took place shortly thereafter, we had a focus on how do we keep our people safe. And then now it's all about innovation and economic competitiveness. And so part of the reason why I'm here today is to talk to you all about your ideas about innovation, tell you a little bit about what's going on at PNNL and the other national labs, and figure out ways where we can better translate the technologies that we're developing inside the labs out into the hands of people like you. So to give you a sense of the scale that we're talking about with the National Lab Research Enterprise, we have about 58,000 full-time staff and over 20,000 scientists and engineers working in the complex. We have 115 Nobel laureates. We have about a little under $14 billion worth of funding, from 82% of which is from DOE, about 11,000 peer-reviewed publications every year. And then this might be of particular interest, 577 commercialized technologies out of the labs and over 6,000 active technology licenses. So I hope that gives you a sense of the scope and scale of the research enterprise within the DOE National Lab System. And now I'm going to shift gears a little bit. Over the next, I would guess, 40-ish minutes, I'm going to be giving you a whirlwind tour of those top 10. I didn't do a countdown because I didn't want to rate the significance of these scientific discoveries. But, Stan?
Are these your top 10?
So they are my top 10.
Okay.
So, and it's basically don't attribute, you have 75 in a brochure actually that I brought. So if you don't like my 10, you can pick from the other 65 that are featured in the brochure. How's that? So, but these were 10 that resonated the most with me, and I wanted to tell you a little bit about them. Like I said, if you're curious about other accomplishments, you can go to the back and grab a brochure. The first one I wanted to talk about was decoding DNA. And so, how many of you have heard of the Human Genome Project? Good, okay, excellent. How many of you know that the Human Genome Project was conceived by DOE? Yes, and hence there's the problem. I know that the two of you are connected to the labs, so you know the story, but nobody else appears to know the story. So yes, the DOE was in fact the agency that conceived of the Human Genome Project. And you may say, why would the DOE be thinking about the human genome? Well, the DOE at the time, this is the late 80s, was really interested in understanding how DNA can change, mutate as a result of ionizing radiation. And they wanted to know what the health impacts of those genetic changes would be. And so they had a radiation biology program that ran for a long time. And so they thought that understanding or mapping and sequencing the entire human genome from a representative human cell would be a way for them to start establishing an understanding of the full genome sequence and what perturbations from that baseline would look like. Now, of course, because this was touching human health, NIH got into the picture pretty quickly. And so DOE and NIH pulled together a memorandum of understanding and jointly initiated the project in 1990. Now, at the time, we did not have the amazing sequencing technologies that we have available at our fingertips today. So this project was originally conceived to run for 15 years and cost $3 billion. Ultimately, and people argue about how long this took, but a generally accepted length of time for the Human Genome Project was 13 years and $1 billion. And part of the reason why it finished early and well under budget was the result of amazing advances in sequencing technologies that took place as a result of that program. The first working draft of the genome was announced in 2000, and papers were published in February of 2001 with the draft sequence. A more complete draft was published in 2003, and that's usually used as the 13-year benchmark. But they continued to sort of finish the genome well beyond that point. And so how much do you think it costs now to sequence a human genome?
$15.
Okay, I like the aggressive nature of this crowd. So without all of the extra stuff like overheads and labor and
The $1,000 $91.
Okay, Stan has got it right. So these days it costs between $1,000 and $1,200 to get a genome sequenced. And if it's considered to be urgent, you can do it in 7 to 10 days. An amazing improvement over $1 billion in 13 years. And if you look at the charts, you can get your hands on these charts, which talk about the cost per kilobase of DNA sequenced and the cost, the time. Both of them are exponentially decreasing plots that are well exceeding Moore's law in terms of the rate that technology has improved. And so this has transformed really biology as we know it. I would like to point out that a team from the Rady Children's Institute for Genomic Medicine recently got a Guinness Book of World Record for the fastest sequencing of a human genome. Anybody want to guess how quickly they were able to do it in? 2 days. 2 days? I hear 2 days. Yeah, okay, so that's a pretty good guess. Any other guesses? That's pretty darn good. 19 and a half hours. I know. When I read that, I thought, wow, the world has changed around us. And so one of the things I did want to point out is that these days we're not just reading DNA, we're writing it. And I know that this is the group that really probably understands that better than most. So this is a picture of Jennifer Doudna. She's at Lawrence Berkeley National Lab. She is one of the inventors of CRISPR technology for genome editing. And so just as the sequencing, the Human Genome Project, really transformed biology and our ability to now read DNA, CRISPR has the potential. It already is transforming biology and our ability to write it and create sort of our own edited genomes per our design specifications. And so this has a lot of ramifications, both positive and potentially negative. And so the field is grappling with some of the ethical considerations that are involved with the technology, as well as just the pure ability to alter the genomes of plants and animals and microorganisms. And, scarily enough, potentially in the future, humans. So, that was number one. Number two, okay, so, I see a lot of, whoops, this is moving a little bit, okay, there we go. I see a lot of young faces in the crowd. Who here was alive before the internet? Okay, there are fewer and fewer of us that can say that. So I was one of those few, and if you want to talk about another transformational technology, it would be really our ability to communicate with each other over things like the internet. And the national labs played a big role in bringing the World Wide Web to the US. And so let's get into our way back machine, way back to the 1989. So in 1989, Tim Berners-Lee, who was a fellow at CERN, developed an idea for something called a World Wide Web, which he thought of as a network of browsable, hyperlinked documents that contain multimedia content. And so as a result of collaboration in particle physics, a physicist from SLAC named Paul Kuntz went and visited with Tim, and Tim was talking all about, this World Wide Web, and as I was reading through the articles talking about this meeting, Paul apparently was not impressed. He was saying, I don't really care about this stuff, let's talk about physics. And he only really got excited about it when Tim said, well, we can share data, potentially. We could, you could link your next computer up to my next computer, and we could share documents, we could share data with each other, and all of a sudden this got very exciting. So apparently Paul Kuntz came back to the US, and by the December of '91, he had set up the US's first web server. And it was set up basically so that they could transfer this kind of information back and forth between the US and Europe. Tim Berners-Lee created the first browser, and later physicist Tony Johnson at SLAC created another important early browser named Midas. Anybody remember Midas? Okay, I sort of dimly remember Midas back in the day. And so if you're interested in taking a look at what the first web server in the US looked like, you can actually go to Stanford Libraries. It has a place called Stanford Wayback, which provides a version of the archived web assets, of which this is one. So if you want to take a trip back through time, I suggest that you check that out. Okay, so I'm guessing that some of you have heard about fracking. Okay, this is really another transformation, this time in energy. And it has the ability to economically extract natural gas and oil from shale. Has been probably the most transformational event in energy since the OPEC oil embargo in 1973. So DOE has been in the energy game for a long time. As I mentioned, early 70s was when it got involved. And it really began to invest heavily into developing technologies for unconventional gas R&D, and they started those investments in 1976. And as a result of that, actually, the national labs got together with firms including Mitchell Energy, the Gas Research Institute, and other private firms to begin to develop and combine technologies. And this includes hydraulic fracturing, but also underground mapping and directional drilling. And if you take all of those things together and you apply them to extracting natural gas and oil from shale, something magical happens. And so what has happened to us as a country is incredible. Between 2007 and 2014, oil production and gas production have increased 60% in the case of oil and 35% in the case of gas. And it's contributed $100 billion annually to the US economy. This is really reshaping the energy landscape. So some of the sectors in energy, such as coal, have suddenly become a lot less competitive with natural gas. And so we're seeing really the economic reverberations of that restructuring in the sector. But I do want to give you a couple of examples of work that was done by the national labs with these industry partners. The first one was the Eastern Gas Shales Project, which was in the Appalachia Basin in 1979. And so what they did was that they proved that shale rock was rich in natural gas. And they used nitrogen foam to fracture shale formations, which led to a deeper understanding of how natural shale fractures. And then the second one was, I like to say this one because I've got roots in Sandia. So high efficiency drill bits, they're commonly used by oil and gas industry, were originally developed at Sandia National Labs. And it was 30 years ago, they developed these drill bits, and it was actually for the geothermal energy community, which ended up not being, I guess, in a frame of mind to adopt the technology at that point. But the oil and gas industry saw these drill bits and they said, we can use these. And so they're now commonly in use by the industry today. So, okay, I think that was number 3. You guys still hanging with me? All right, okay. Are you impressed yet?
Yes.
Okay, so found life's messenger. This has to do with the story of the discovery of messenger RNA. And so for those of you who maybe aren't immersed in biology, messenger RNA really carries encoded sequences from DNA and provides sort of instructions for the cell to synthesize proteins. So it's really considered to be an intermediate step of what is considered to be the central dogma of biology, which is DNA to RNA to proteins, which we know is a really simplified model, but for the purposes of our discussion, let's just stick with that. Let's see, I'm gonna click and I'm gonna show you a picture of Ken Vulcan. So he was the first person, he and his collaborator was the first person to actually observe messenger RNA in the laboratory. And they weren't originally attributed with this discovery because of the way that they talked about it and they published it. So the way they discovered it is that they saw, basically, after a bacterial cell gets infected by phage, that if they introduced isotope labeling, the isotopes would get incorporated in RNA, sort of after an initial early time point. But beyond that time point, it would get much more heavily incorporated in DNA. And so they interpreted that as, well, RNA must be a precursor to DNA replication. And they called it DNA-like RNA. Several publications came out 3 years after the fact, talking about the observation of mRNA. And in those papers that were published in Nature, the authors attributed a variety of features to the mRNA that they had detected, which actually lined up very nicely with what Vulcan and his colleagues had identified in their DNA-like RNA. And so after the fact, the community has recognized that in 1958, 3 years earlier than what is commonly thought of as the seminal papers on mRNA, it was discovered by a DOE lab scientist. So yay, good for us. Okay. I'm sure that some of you have seen DVDs, CDs, okay? Good. This isn't foreign to you. I think it's transformational. At least I did when I've got a little bit of time on Friday night, box of popcorn, pop a DVD into DVD player. But what a lot of people don't know is that actually optical digital recording technology originated at a DOE lab called PNNL 40 years ago. And so I show a picture here of the inventor at PNNL, Jim Russell. And in his hand, he's holding a 5x7 photographic plate, which contains a half-hour color TV program with quadraphonic sounds. I know.
Technicolor?
Ooh, I know. It's really exciting. So how did Russell kind of clue in to optical digital recording technology? It turns out that he is a very avid music listener, or he was a very avid music listener, and he got frustrated with the wear and tear that record players would create on vinyl records. And he thought, gosh, there's got to be a better way to store sound and have it play back without having to deal with all of this sort of degradation in the sound. And so, again, reading stories about Jim, he said that he was alone at home on a Saturday afternoon when all of a sudden he realized that he could avoid all that wear and tear by using light to read the music instead of a stylus. And so because he was familiar with digital data at the time, he knew he could accomplish this task in a digital way. That would be more efficient and effective. And if he could make the binary code compact enough, he could store not only symphonies, but a huge amount of information, like encyclopedias on a piece of film. And so Battelle, who was an operator at PNNL at the time, still is, he brought his invention forward and was originally met with some skepticism because people apparently told him, how can you possibly encode sound using light? That makes no sense. But to their credit, Battelle eventually did let him pursue the project, and he did, after years of work, succeed in inventing the first digital to optical recording and playback system. He filed a patent in 1966, which was granted in 1970. And so his work forms the foundation, along with several early other pioneers, of the optical digital recording technology that we enjoy in CDs and DVDs and data storage today. Pretty cool. Okay, do we have any chemists in the crowd? Yeah, got some chemistry? Got some chemistry love out there? Okay, good. I think that you would probably then be extra excited to know that the periodic table would be a lot smaller without the national labs. So the national labs have discovered 22 elements, and I have my notes in front of me because I am going to read them to you, and boy, are they a mouthful. So we have technetium, promethium, astatine, neptunium, plutonium, americium, curium, berkelium, californium, Einsteinium, Fermium, Mendelevium, Nobelium, Lawrencium, Rutherfordium, Dubnium, Seaborgium, Flarevovium, Muscovium, Livermorium. Go wonder who discovered that one. Tennessine, that must have come from Oak Ridge, and Oganesson. Okay, so a few little highlights. Technetium has really revolutionized the field of medical imaging, and who knows which one of these 22 is in your smoke detectors? Americium, thank you. What was that? An alpha emitter. Do you want to come up and give the talk? I mean, that's good. Okay. So, how many of you guys have done the PNNL salute? Okay. Rick knows what I'm talking about. Yeah, he and I were swapping some stories about doing the PNNL salute. So if you've gone through an airport, more than likely you have done a PNNL salute. And so in this picture, we have Secretary of Energy, Rick Perry, doing the PNNL salute along with our lab director and other friends. And behind them, they have a millimeter wave scanner. And so millimeter wave technology was invented at PNNL. And this technology passes harmless electromagnetic waves over a human body. And the waves that get reflected actually are converted into a 3D image in a computer. And so it's like a fuzzy photo negative. Not to worry, the signals are 10,000 times less powerful than a cell phone signal, and they don't penetrate the skin. This is a great example of technology that has come out of a national lab and has gone into the marketplace, and so I would love to see more successes like this. In 2003, the technology was introduced with a partner, SafeView, as the SafeView Scout system for security applications. And in 2006, L3 Communications acquired SafeView, and they have rebranded and renamed the millimeter wave whole body imager as the ProVision. And so to give you a sense of how much this has changed the way that we walk through our airports, it was originally deployed at Los Angeles International Airport in April 2008. And so since then, nearly 250 millimeter wave devices have been installed in US airports, and 600 have been installed worldwide. And the TSA plans to deploy about 1,000 body scanners at US airports, about half of which are utilizing these technologies. When people tell me I shouldn't talk about the PNNL salute, you know, because people are like, I don't really want to do that in an airport, I always say, well, you could always get up close and personal with your friendly TSA agent. That's an alternative, right? And usually people go, okay, all right. But what is also interesting about millimeter wave technology is that it can be used for a variety of other purposes. And so, you know, things that we've been thinking about at the lab is, can you use it to develop custom safety equipment? Can you use it for ergonomic applications? Okay, who's seen Ready Player One? Okay, so you can use it to actually develop a 3D body scan and use it in virtual gaming, develop an avatar. You can use it to detect objects concealed in walls, and it's been licensed actually to IntelliFit, which is a company that is focused on consumer measurement technologies for body measurement applications. So if you want to have your body measured, I guess go and look at IntelliFit. So anyway, moving on. So revealing the secrets of matter. Now we're wandering into really deep science explorations that are being done by the national labs. So protons and neutrons, when I was in school, they were taught to me as these sort of elemental particles they were indivisible. They were the smallest things that you can think of. Well, what we've discovered as a result of many of the particle physics experiments running around the world, sponsored by DOE, is that actually there are more elementary particles than the protons and neutrons. And so, you know, we have quarks, we have neutrinos, we have the Higgs particle. And so this has really transformed the way we think about matter and how the material world works. I'll just focus in on the Higgs particle example. So at CERN, researchers look for the Higgs boson at the Large Hadron Collider by accelerating protons to high energies and then crashing them into each other. And they look at the energy and how that energy converts into mass, and they look at the cascade of particles that get generated as a result of the collision. And that tells you a little bit about what particles actually are comprising those protons. And so Higgs bosons, they are only produced in roughly 1 out of 1 billion of these collisions. And they live only 1/septillionth of a second before their energy is converted into a cascade of other particles. And so the scientists have been studying these secondary particles to study the Higgs properties. And in July 4th, 2012, 2 experiments at the Large Hadron Collider announced that they each observed a new particle consistent with the Higgs boson, particle which is predicted by the standard model. And in August 2018, they finally have observed the Higgs boson decaying to bottom quarks. Now, I'm sorry, I'm not a particle physicist, so while I think that this is super cool, if you tell me what the difference between you ask me what the difference between a strange quark and a charm quark is, I wouldn't be able to tell you, so please don't ask me any questions. But I am very impressed at the fact that they are getting to the fundamentals of the building blocks of matter, and that tells us a lot about how our universe is constructed at the subatomic level. Okay, we're getting close to the end. I know I'm hitting you guys with lots of science here. But I do want to talk about the world's smartest computer. So you may have noticed that there was a little factoid on my slide about the National Lab Complex and all the people who work there and all the funding that you have. 32 of the world's fastest supercomputers in the world are DOE computers. And so in June 2018, the US took back world dominance in exascale computing with the launch of a new machine called Summit at Oak Ridge National Lab. And the DOE partnered with IBM and NVIDIA to construct Summit. It has a peak performance of 200,000 trillion calculations per second, or 20 petaflops. It's capable for some applications of more than 3 billion mixed precision calculations per second, or 3.3 exaops, okay? So that's pretty impressive, but it also has more than 10 petabytes of memory that's paired with fast, high bandwidth pathways for efficient data transfer. And it's an evolution of this hybrid GPU, CPU hardware architecture that was pioneered by Titan, which is a 27 petaflops DOE computer in 2012. And so we're now, the national labs and researchers around the world have this as a resource for us to do calculations at an unprecedented scale in applications ranging from energy to advanced materials and artificial intelligence, amongst many other things. Okay, last one. Number 10, shed light on photosynthesis. So photosynthesis is the process by which plants convert carbon dioxide into glucose, and it's a very complex biochemical set of pathways. Dr. Marvin Kelvin, he spent many years trying to trace the path of carbon through the photosynthetic pathway. And the way he did this is that he used carbon-14 as a trace to label carbon dioxide. And after adding this labeled carbon dioxide to an illuminated suspension of green algae, he stopped the algal growth after certain time points, and he used paper chromatography to isolate and identify each species that had the radio-labeled components in it. And so what this enabled him to do, this painstaking, I can't even imagine it, this painstaking research enabled him to trace down most of the chemical reactions in the intermediate steps of photosynthesis. And as a result, he was able to define what we call the Calvin cycle in biology. This was the first use of a carbon-14 tracer to explain a chemical pathway, and he won the Nobel Prize for Chemistry in 1961. So, okay, are we all impressed? This is a picture of him, by the way. So I just want to end by saying I know I hit you with a lot of stories in rapid fire succession. But we're really, I think I talked a bit about how fields of science are transforming at an extremely rapid rate. Computing is one of them. Biology is another one that I talked about. We have sectors that we're working in that are also changing very quickly. Energy is one. Cybersecurity is another. And so it's really important for the whole research community, not just the national labs, to be thinking towards the horizon, which is why I put this slide up here. Some of the initiatives that we're leaning into right now are in artificial intelligence and machine learning. We're looking at quantum, sort of ranging from quantum computing and quantum materials, as well as detectors for quantum events. We are very closely following what I had mentioned in my first slide about biology and the transformational technologies, particularly CRISPR, genome editing. But we're at a very exciting time in science overall. For those, there have been people who've said, we've learned everything there is to learn. I am one of those people who think that we've barely scratched the surface of what there is to know about our planet and the universe. So I thank you for joining me here tonight. I thank you for listening to these 10 stories. And I hope you walk away with a greater appreciation for the kind of work that goes on at your friendly neighborhood national lab. Thank you.